Good morning, good afternoon, and good evening, everyone, from wherever you're joining us. And welcome to today's webinar titled Network Meta-Analysis and its Role in Evidence-Based Decision Making. Today, three experts will provide an in-depth exploration of NMA methodologies, practical applications, and real-world impacts. I'd like to thank the sponsor of today's event, ICON, for making it all possible. I would like to introduce our speakers today. Ankit Pawa is a lead epidemiologist at ICON and has 16 years of experience in statistical modeling, analytics, and programming. Daniel Gallardo is a senior consultant in health economics and epidemiology at ICON and brings deep expertise in health technology assessments and meta-analysis methods. Nathan Green is a senior research fellow in the Department of Statistical Science at University College London, and his research interests include health economics, survival analysis, evidence synthesis, and epidemiology. I am going to bring our first speaker on here. Hi, Ankit. Thank you so much for being here with us today, and please take it away whenever you're ready.
Yes, thank you, Sarah. Welcome, everyone. In today's session, we'll explore the fundamentals and practical applications of network meta-analysis, or NMA, and its growing role in evidence-based decision-making. To begin, I'll walk you through the basic concepts of NMA to help build a solid foundation. Then we will discuss the importance of NMA in healthcare and particularly how it supports more informed and transparent decisions when multiple treatments are involved. I will also cover an overview of NMA methodology, how it works, and what makes it distinct. After that, Nathan will take over. He will provide insights into how health technology assessment bodies view NMA. He will introduce you to relevant HTA guidelines, and then he will also touch base on some advanced evidence synthesis methods such as matching adjusted indirect comparison and multi-level network meta-regression. Finally, my colleague Daniel will guide us through the practical sides of things. He will show implementation of NMA using R programming. He will cover some key R packages like NetMeta and MultiNMA and show a live demo for you today. And then I'll return at the end with some concluding remarks to wrap things up and tie it all together. So let's get started. Okay, so to understand how network meta-analysis fits into the broader landscape, it's helpful to start with a taxonomy or classification of different types of comparisons used in evidence synthesis. First, we have the direct comparison, also called head-to-head evidence. Here, two treatments are compared within the same study or a trial. Next, we encounter the naive or unadjusted indirect comparison. This involves comparing absolute treatment effects using outcomes from separate trials without accounting for differences between study populations. It's simple but prone to bias. Then we have adjusted indirect comparison, and it's commonly referred to as the Booher method. This approach compares relative treatment effects while adjusting for a common comparator. Typically, that comparator is a placebo or a standard of care. And finally, we arrive at network meta-analysis, also called a mixed treatment comparison. And this method extends adjusted indirect comparisons to networks involving multiple interventions. NMA combines both direct and indirect evidence from a network of studies, and that generates more comprehensive and connected treatment comparisons. So here's an example with which things will become more comprehensive. On the left, you'll see a list of identified studies that provide direct comparisons between various treatments and placebo. There are three types of direct comparisons possible here. For example, we can compare drug A versus placebo using the first two studies through traditional pairwise meta analysis. Similarly, we can analyze the effects of drug B and drug C versus placebo by pooling results from the relevant studies. But what if we want to compare drug A with drug B or drug A with drug C when no direct head to head studies exist? Well, that's where indirect comparisons come in. Using the Bucher method, we leverage the common comparator, that's placebo in this case, to derive relative comparisons between treatments that haven't been directly tested against each other. Let's quickly go over how a network of evidence gets built. Mixed treatment comparison, or MTC, generalizes standard pairwise meta analysis to networks involving three or more treatments. On the left side of the slide, we have multiple two-arm and three-arm studies, trials in this example, and these are arranged by which treatments they compare. On the right, this information is visualized as a network diagram. Each node represents a treatment, and each line or edge represents a comparison supported by trial data. When treatments are connected through multiple direct or indirect comparisons, closed loops form and allow us to conduct an MTC. This network of evidence is a crucial tool. It not only shows how well connected the available evidence is, but it also helps assess the feasibility and robustness of conducting NMA. So before we get into the feasibility of conducting an MTC and NMA methods, let's first go over the importance of NMA in healthcare decision making. Meta analysis, when appropriately applied, is a powerful statistical tool that helps us make some informed conclusions by combining results from multiple studies. First, it increases statistical power by pooling data from separate studies, allowing to detect treatment effects that individual studies do not uh, they do not show, or due to some small sample sizes. Second, it improves the precision of effect estimates. By combining evidence across studies, we reduce uncertainty and obtain narrower confidence intervals for treatment effects. Third, meta analysis allows us to answer questions not addressed in individual studies. So, as an example, we can investigate consistency across subgroups or assess variability in treatment performance across settings. It also enables us to generate new hypotheses. Um, Unexpected findings or variations across studies may highlight areas for further research or refinement of clinical practice. Importantly, in the context of evidence based medicine, meta analysis plays a key role in synthesizing totality of available data, helping guide clinical decisions, policy making, and resource allocation. And when we move from pairwise to network meta analyses, these benefits are expanded further, allowing for comparisons across multiple treatments even when direct evidence is incomplete or missing altogether. As I said, network meta analysis has become an essential tool in healthcare decision making, particularly when comparing multiple treatment options. And unlike traditional pairwise meta analysis, NMA allows simultaneous comparisons of several treatments, even when some head to head trials are missing. This is particularly valuable in real world contexts where not all comparisons are available. By synthesizing both direct and indirect evidence, NMA strengthens evidence based decision making, offering, offering a more complete and connected view of treatment performance. This helps clinicians and policymakers understand which treatments are most effective or safest across a broader therapeutic landscape. NMA also plays a critical role in cost effectiveness analysis and resource planning. It helps identify treatments that offer the best value for money, and that's crucial in settings where limited healthcare budgets are available. These analyses are often used to inform clinical guidelines and policy decisions, with many HTA bodies and professional societies relying on NMA results to support their recommendations. Also, NMA promotes methodological rigor and transparency. The approach follows structured process, that's systematic review, network construction, then analysis, and then treatment ranking, making results reproducible and auditable. As shown in the flowchart on the right hand side of the slide, the process involves steps of literature search, followed by evidence synthesis, and that leverages treatment ranking, which helps with informed decision making. Overall, NMA supports better clinical, economic, and policy decisions, especially when multiple treatment pathways need to be evaluated side by side. Before conducting MTC, it's important to assess whether the available data support this approach. This starts after completing a systematic or targeted literature review, as mentioned on the previous slide. So, and then we begin by examining whether the included studies provide both direct and indirect evidence. So, if so, we evaluate the feasibility of conducting MTC. The first step is to ask, do the patient populations across studies match the population relevant for decision making? If all trials are sufficiently similar, then can be then they can be pooled directly in an MTC. If only a subset of study meet this criteria, we check whether there's evidence of effect modification, meaning that differences in treatment effects are due to population or design characteristics. But if no major differences are expected, we can still include these studies in the MTC. However, if significant heterogeneity is present, we either restrict the analysis to the subset of similar studies or use meta-regression technique to adjust for known sources of variability. This feasibility assessment is crucial for ensuring that the evidence synthesis is appropriate, robust, and interpretable, ultimately leading to valid treatment comparisons. Now, there are two main statistical paradigms used in network meta-analysis, that's frequentist and Bayesian. Among the frequentist methods, the Bayesian method is widely used for adjusted indirect comparisons. It's based on simple equations and it is acceptable by regulatory bodies like Australia's PBAC and Germany's HTA body. So, my colleague Nathan is shortly going to talk about HD guidelines on anime in more details. For larger and more complex treatment networks, a frequentist regression based approach, such as weighted least square regression, is recommended. This approach has been formalized by Rucker and his colleagues and allows for efficient synthesis of direct and indirect evidence. On the other hand, Bayesian methods offer several advantages, particularly in terms of interpretability and flexibility. For example, UK's NICE Decision Support Unit, the DSU, has provided extensive guidelines on using Bayesian anime methods. Nathan will touch upon this as well. The choice between frequentist and Bayesian approaches often depends on regulatory expectations, network complexity, data sparsity, and desired form of interpretation and inference. Both methods are valid but differ in philosophy and their outputs. So, frequentist regression based NMA is grounded in classical statistical theory. It relies on concepts of repeated sampling and focusing on hypotheses testing. As mentioned earlier, this method uses weighted least square regression to estimate relative treatment effects. The weights are derived from inverse of the variance, ensuring that more precise studies contribute more to the uh, pooled estimate. Results are typically presented as forest plots showing point estimate and 95% confidence intervals. However, it's important to note that these confidence intervals cannot be interpreted probabilistically. So, a 95% confidence interval means that if we were to repeat the study 100 times, the true value would fall within that range 95 times. Frequentist NMA does not produce direct probability statements about treatment effectiveness. Instead, it assesses whether differences are statistically significant or not. But when it comes to treatment ranking, frequentist methods use the P score, which is analogous to the Bayesian SUCRA that I'm going to talk about on the next slide. So the P score estimate the extent of certainty that one treatment is better than others based on the point estimate and standard errors. Overall, this approach is computationally efficient and aligns with HTA expectations in many jurisdictions. However, it has interpretation limitations, particularly around confidence intervals and ranking probabilities, which may require complementary analysis or clear explanation for non technical stakeholders. Okay, so the Bayesian NMA offers several distinct advantages, especially in complex decision making scenarios. First, it enables intuitive treatment ranking through surface under the cumulative ranking curve, that's SUCRA. Unlike frequentist methods, Bayesian approaches generate posterior probabilities that a treatment is better than a comparator. For example, if a posterior probability is greater than 85%, it suggests the treatment is likely to perform better. If probability is less than or equal to 15%, it's unlikely to perform better. This probabilistic output helps stakeholders interpret results with more confidence. Bayesian models also allow covariates to be included via meta regression, especially useful when there are 10 or more studies. This enables assessment of effect modification and exploration of heterogeneity. Another major benefit is the interpretation of the credible intervals. A 95% credible interval means that it is 95% prob- that there is a 95% probability that the true effect lies within the interval, a more intuitive interpretation than frequentist confidence intervals. Bayesian NMA does not rely on hypothesis testing. Instead, it provides posterior distributions for each treatment effect, making it well suited for decision making under uncertainty. Finally, in situations with sparse data, for instance, fewer than 5 studies, Bayesian methods can incorporate informative priors on between study heterogeneity, making better use of limited evidence. And altogether, Bayesian NMA is powerful, flexible approach, particularly valuable for transparent decision making and stakeholder communication. So, in any meta analysis, including NMA, it's inevitable that the included evidence will differ and in various ways. This variability among studies is known as heterogeneity. And there are three main types. The clinical heterogeneity, it arises from differences in patient populations, interventions, dosages, comparators, or outcomes. Methodological heterogeneity, it refers to variation in study design, such as blinding, allocation methods, or risk of bias. And then statistical heterogeneity. This is the variation in effect estimates that results from clinical and methodological differences. This is usually assessed using statistical measures such as the I-square statistic or the between study variance tau square. Understanding and addressing heterogeneity is essential for drawing valid conclusions from a network meta-analysis. When heterogeneity is minimal, results are more consistent and generalizable. However, when heterogeneity is substantial, we must either explore it using subgroup analysis or meta-regression or adjust the model strategy. For example, switch from fixed effects to random effects model. And I'm going to talk about that in the next slide, the last slide. Proper assessment of heterogeneity enhances credibility, interpretability, and policy relevance for your findings. It's not about eliminating differences, but about accounting for them systematically. So, choosing between fixed effects and random effects model is a key step in NMA. In a fixed effects model, we assume that all studies are functionally identical and are estimating the same underlying effect size. This approach is appropriate when there's minimal variability among studies and when the objective is to estimate a common effect for a specific population. On contrast, a random effects model assumes that the true effect varies across studies due to differences in population, their settings, or other factors. Here we estimate distribution of effect size, including both a mean and a standard deviation. Consider this. A pharmaceutical company conducting multiple trials on the same drug may justifiably use fixed effects models since these study designs are standardized. However, when pooling data from independent published trials, especially those with difference in PCOS elements, that is, population, intervention, comparator, and outcomes, a random effect model is more realistic. Statistically, fixed effect models yield narrower confidence intervals, assuming no heterogeneity, and random effect models produce wider intervals reflecting greater uncertainty but also provide more generalizable results. Also, in fixed effects model, larger studies may dominate the results, while in random effects model, smaller studies could carry substantial weight since the model accounts for between study variation. Ultimately, the choice should be based on your understanding of the clinical context and data heterogeneity, not just statistical tests.
So Ankit's just done a great job introducing NMA. I'm going to sort of build on that, but I thought it would help just to first show the landscape of how NMA is used and how it's considered in different HJ bodies. So some of these are kind of my summaries, so kind of my opinions, but to give a feel like that, for example, here in the UK, NICE, well, Ankit's mentioned the decision support unit documents, excellent technical methodological guidance, and they have with them Bayesian code, so they are very amenable to more sophisticated modern models, often Bayesian rather than frequentist. Whereas like the contrary to that is somewhere like Germany, which can be a little bit more reserved, and I said cautious here, and perhaps they would not prefer a Bayesian model, and maybe they would prefer a model which would not incorporate indirect evidence in favor of direct evidence. So there's different preferences depending on different localities. And I have a map here just to sort of summarize that. So some of these more conservative areas, perhaps Australia and Germany, and maybe more amenable to maybe more modern, maybe less tested methods, including Bayesian and MLNMR methods, maybe with Canada and United Kingdom. So it does vary. I'm going to be talking about some of these newer methods, but the first thing to say is why these exist. So Angus just talked about NMA, but there's an assumption in here that that the effect modifier variables are balanced across studies here, which may well not be the case, and that would bias the results. So that is to say that maybe the cohort, the characteristics of the individual in different trials may be different, and that's important because it would change the effect, the relative treatment effect. So we need to account for that. An earlier approach for this were the what's called MAIC and STC, so I'm going to talk about those first, and then MLNMR afterwards. So you can kind of think of these methods coming into two groups, really. One of them is kind of a weighting approach, and the other one is a regression approach. So the idea is to re-weight one study so it looks like another study, and another is to fit a regression model against the study, against one of those studies. So the first method is matching adjusted indirect comparison, which is probably the most popular type of weighting approach. And I've put some equations on here, but not to be sidetracked by them. But it's to essentially use an optimizer or method of moments, if you're aware of that, and kind of match the covariates between two trials. So the idea here is that you have two trials, whereas one of those trials you have the individual level data, perhaps your own trial, whereas you don't have individual level for other trials, for competitors. You only have aggregate level data. So that may well be from publications or other openly available sources. So the idea is that you can somehow sort of recalibrate using the aggregate level data, the distribution of your covariates in your individual level data. So that's essentially what's happening here. We're reweighting the individual level data so it looks like the aggregate level data. And that obviously brings us a restriction there, because the target population always has to be the aggregate level data. So if we wish to have a different target distribution of the covariates, well, that's not possible using something like MAIC. There's also the issue of how well these two studies, the aggregate and individual level studies, match one another. So if we wish to reweight one of them to look like the other, we need a certain degree of overlap. And without that amount of overlap, this approach can particularly struggle. And this comes into ideas where the regression approach maybe is better suited. If there is not a lot of overlap, this can result in things like poor, what's called effective sample size, whereas maybe only a few individuals in the individual level study actually account for most of the weight. So you can imagine that if you're counting on just a few people for all of your effective sample size, that can cause some problems. The second approach I mentioned is something called simulated treatment comparison, and this is the regression approach. And so the idea here is that we fit a simple regression to the individual level data trial, and then we predict using that fitted model, using the covariates from the aggregate level data. So the simplest approach is what's called a plug-in method, where the covariates that you plug in would be these summary statistics that we get from papers, for example, and then we forward predict using those means and standard deviations, and that brings with it issues with bias, for example, when these are nonlinear functions. More modern ways of doing this is to forward predict using an actual distribution of covariates, which is sort of derived from the summary statistics. And this is something closer to G computation, which I'll come to. So both these approaches, they have their limitations. Some of them I've already mentioned. So you have to consider that you are constrained by the target population being from the aggregate level study. These approaches are only designed for pairwise comparisons, and obviously we've been looking at NMA up to this point. So, not surprisingly, this idea that's encapsulated in multi-level network meta-regression, a name I still don't like, is to address some of those issues. So the idea here is that we can take this situation where we have our own individual level data and some other aggregate level data, but we can extend this idea. So MLNMR can take not just 2 studies, it can take any number of studies, and it can combine those into a network of evidence like we've seen in NMA, and we can do that sort of simultaneous evidence synthesis on all of those studies at once rather than pairwise. This is an idea that was originally published by Jackson et al. In something called ecological regression, and there were earlier results just looking at discrete distributions, and then this has been extended and excellent software implemented by David Filippo, and Daniel will come to that later. So the thing that I want you to take away from MLNMR, it's essentially a 2-step process. The first is to define the individual level regression, a bit like the outcome regression in the STC. And the second step, which is the clever part, is that we average or we integrate over this aggregate study population to form an aggregate level model. And in that way, we can combine the aggregate level model and the individual level data into a single model. So that looks something like here. So on the left here, we've just got our standard regression on our predicted linear scale. And this could be like number of counts or died or not died or whatever, but at the individual level data. And then on here, we've got the aggregate level data, which would be not our data from publications, for example. And that's indicated here by this dot here, where it's kind of like an average or a sum. And the clever part here is where we integrate over this individual level regression equation in here, and we integrate over the distribution of covariate in the aggregate level data studies. And in that way, we can link these 2 models via this eta function here. So extremely powerful and very flexible. Like I said, you can use any number of studies, and it can be quite straightforward depending on the type of data you have, but also very flexible to different types of data. So this approach is, like I said, it's pretty new, but it's really been an open-ended research. It's really moving quite quickly. So issues to do, I should probably go back, issues to do with where we get this F distribution from, the distribution of the aggregate level data. There are different alternatives to that, and also how we can do this integration. In some cases, that's relatively straightforward. We can actually write that down. In other places, we would have to resort to numerical integration methods. So there are still open questions to do with MLNMR. Some of those are when we have unanchored networks. So that's when we don't have a common treatment. Until recently, the survival analysis models with survival data, that wasn't implemented, but now that's newly implemented, published work and implemented in the multi-NMA package, which is great. There's also issues about whether or not we're interested in the conditional or the marginal effect. So that's whether or not we want the effect for particular values of covariates, or whether we want the relative effect for kind of averaged over a population. And that will depend on your particular problem, and arguably when you're doing something like cost effectiveness analysis, health economics, a marginal effect may be more appropriate. So this table is just to summarize some of these pros and cons, which I've been talking about already. The SC MAIC approaches are definitely more mature and prevalent, but the MLNMR is really like coming up behind strongly from the pack, and I think it will only be in more and more use as time goes on. However, if you go back to my first slide with the map, something like MLNMR may well not be as preferred, if you like, in certain jurisdictions, certain bodies, because perhaps of its novelty and complexity, but I can well imagine things changing over time. Finally, there are further extensions to these models, depending on your particular problem. So I've put a few down here which I've worked on. So there's something here, model-based NMA, which is when you might have studies which have results at different dose levels, and you can still incorporate inside an NMA the additional information of different sizes of dose. You can also incorporate subgroup analyses into the network meta regression, and that can be one way around of some of the issues that you can have with using, for example, MLNMR. And then finally, I did mention G computation with reference to STC. There are some very novel extensions to the STC approach, which are better at handling the uncertainties in the model, and some of the issues that you would have in terms of the working out the marginal rather than the conditional. So this really is a very exciting area, and I'm sure there's probably lots of current work which I'm not aware of, but it's something which will only become more and more prevalent and kind of more and more powerful as time goes on. So that's kind of my overview of these novel methods. And now I'd like to hand over to Daniel to talk about how you can actually implement these things.
Okay, thanks, Nathan. Okay, before moving on to the fun stuff, some notes about how we present meta-analysis results. So meta-analysis results are typically reported both graphically and numerically. You will often see, just as Anki said, the most iconic plot in meta-analysis is the forest plot, which visually summarizes the findings from each individual study along with the overall pool estimate. Effect measures depend on the outcome type. So, for example, for binary outcome, we use odds ratio or relative risks. For continuous outcomes, we use the difference in means, either raw or standardized if scales differ. For time to event outcomes, we typically report hazard ratios. But each study's influence on the pool estimate is based on inverse variance weighting. So that said, larger, more precise studies will carry more weight into the analysis. It's usual to accompany forest plots with raw data such as sample sizes and event counts, for example, for binary outcomes. But the key summary is the pool estimate with its associated uncertainty presented as 95% confidence interval in the frequentist approach, or 95% credible intervals in the Bayesian framework. These estimates tell us whether the intervention of interest is better than the comparator from a statistical point of view. And to ensure studies comparability, important covariates like patients' characteristics may also be presented. And finally, between study heterogeneity is routinely assessed using statistics like the I-square or the Q statistic. So now let's move on to of the most widely used R packages for conducting network meta-analysis. We're going to see NetMeta and multi-NMA. So we will follow the same structure for both, starting from with a high-level workflow overview, then walking through each step in more detail. So this slide shows the workflow of the NetMeta package, in which we firstly import the data. You can import your own data, or you can import a predefined data within the package. You conduct a bit of data wrangling, then the main analysis. For example, we are going to conduct such standard network meta-analysis. Then we're going to conduct some relative effects computations and present some data visualizations. And finally, we go over the model evaluation based on inconsistency and heterogeneity. So a key point to keep in mind is that NetMeta requires contrast-based data. meaning each study reports treatment effect estimates based on direct comparison between arms. Okay, in two studies, this is quite straightforward, but for multi-arm studies, you need to include all pairwise comparisons, ensuring consistent study levels across all entries of the same study. To go through some of the most important functionalities of this package, we are going to use this study that is STOW 2010, which investigated the mean off-time reduction in patients giving dopamine agonists as adjunct therapy in Parkinson's disease. We are going to use this dataset in that case and for the multi-NMA package also. To meet all the package requirements previously commented, we have to run this line of code, and we will obtain something like the data frame you can see in the slide, and we're going to present all the variables in the next slide. So, okay, once the data is prepared, we can run the network meta-analysis using the core function that it's netmeta. And as you can see, key arguments include the treatment effects, standard errors, the treatment levels for the active treatment versus the comparator, the study identifiers, and the data frame containing all this data. In this case, as we set common false, we are going to conduct a random effects model. And we set plus as placebo as the reference treatment. So after running the model, we print the output and give us some characteristics of the model, like number of studies, comparisons, number of patients, number of designs. Here, a design refers to a set of treatments compared within a trial. So, for example, a design could be treatment A versus treatment B, or treatment A versus treatment B versus treatment C. That could be separate different designs. Treatment effects are then shown relative to placebo, which is why placebo itself has no estimate. And below we see some heterogeneity estimates, but an important thing here is the breakdown that at the final part of the output into within design variation and between design inconsistency. And for example, in this case, most of the heterogeneity is within designs, meaning that even studies comparing the same treatments show real differences in effect sizes. In network meta-analysis, it's common to report a set of data visualizations, as my colleagues commented. So first, the network graph helps us see which treatments were directly compared. In that case, nodes are scaled by sample size, for example, and the edges, the line thickness reflects the number of studies. So for example, here we have 15 comparisons between dopamine agonists and placebo. Next, we see forest plots, and we perform two types of forest plots. The first one, we show the effects relative to placebo, and in the other one, in the right hand, the other one shows all pairwise comparisons among active treatments. And finally, in the top right on the slide, we can see the rankogram. This plot ranks treatments by effectiveness using the frequentist p-score. So we find, for example, in that case, that dopamine agonists rank the highest. Okay, our next step will be model evaluation regarding heterogeneity and inconsistency in the network. So we start by feeding what is called a design by treatment interaction model to check for overall inconsistency. The key takeaway is in the final section of the output that there's no evidence of inconsistency between designs. To go further, we use the side method in the net split function, which stands for adequate, separate indirect and direct evidence, which compares direct and indirect evidence for each pairwise estimate. For example, in our case, we have 3 comparisons that have both types of evidence, and none showed inconsistency because the p-score is above 0.05. Lastly, we can generate also a net heat plot to visualize which parts of the network contribute to inconsistency. But again, the heat plot indicates no major disagreement between direct and indirect comparisons in that case. Okay, so let's shift from our frequentist vision of the world to the Bayesian and look at the multi-NMA package. Okay, As shown in this slide, the overall workflow is quite similar to NetMeta, but multi-NMA offers, in our view, additional flexibility, for example, including support for meta-regression and multi-level network meta-regression models. So first, we will set up our network. You can directly import this data set from the package, and you will notice that the format of the data is different compared to the one we saw in the NetMeta package. This is long format data that is 1 row per study arm. In this package, we can use arm-based, contrast-based data, or even a combination of both types of data. For instance, we use arm-based data to build our network, and we can see information about the network related with the number of studies, the number of study arms of each study, the design of each study, and so on. And the final part shows if our network is connected or disconnected. Related with this connectedness of the network, unfortunately, we don't cover more complex topics like how to handle sparse networks, for example. But if you are interested, we can briefly discuss it later. Okay, that's the network plot from the multi-NMA package. And we set in weight edges and weight nodes argument to true. We weight the nodes and the edges from our evidence plot. Okay, so once the network is defined, a recommending strategy is to fit both fixed and random effects model using multi-NMA and using NMA function, it's the core function. One key difference that you will notice is in Bayesian modeling is the use of priors, as my colleagues commented. The priors guide the estimation of posterior distribution of the model parameters. So in our case, for example, we apply weekly informative priors, allowing the data to primarily drive the results. The model output includes details such as the likelihood that we use, the link function, the number of Markov chains, number of iterations within each chain, and so on. But before even start interpreting the results, it is critical to check the potential scale reduction factor, that is this R hat parameter. That should be lower than 1.05 or 1.1, depending on the reference, but meaning that the model reached the convergence. And the T parameters represent the treatment effects indexed by the treatment number. To assess the model fit, we use model fit parameters like the deviance information criterion, or DIC, and residual deviance. As a rule of thumb, lower DIC indicates better fits. For residual deviance values, close to the number of data points suggests a good fit, since each data point is expected to contribute to about 1. So based on this criteria, we see that the DIC values of both models are so close, even the residual deviance. So based on that criteria and aiming for parsimony, that it's the selection of the simplest model, we select the fixed effect model in that case. And we can also plot the residual deviance contribution for each study, for each data point. So the multi-NMA package let us assess inconsistency at both the global and local levels. For the global inconsistency, we can fit what is called an unrelated mean effects model, which removes all the consistency constraints. It's just like conduct separate pairwise meta-analysis. And when we have the two models, we can compare it to the standard model using a depth plot, that stands for deviance plot, which shows how each data point contributes to model fit. In that case, roughly all the points fall along the line of equality that we can see. So that means that there's no evidence of inconsistency. And for local inconsistency, we use not splitting method, that is just like in the frequentist approach. Once we have selected the best model, we can visualize the prior and posterior distributions of key parameters, including, in that case, for example, study-specific intercepts or relative treatment effects. And we also get the relative effects compared to the reference treatment that we set that our reference treatment would be the number 1, along with the full distributions. We can plot also these full distributions. A powerful feature of this package that I think it's worthy to mention, and it's a bit beyond the scope of this webinar, but it's its ability to generate absolute treatment effect predictions by assuming a distribution for outcomes under the reference treatment. I think that it's quite powerful. And finally, treatment rankings, but also rank probabilities and cumulative rank probabilities can be computed to visualize the probability of a given treatment being more beneficial than the rest. And yeah, now I hand over to Hankit, who will wrap up some take-home ideas.
Thank you, Nathan. I think it was great presentation. Okay, and thank you all for staying with us through the session. And to wrap things up, I'll quickly summarize some key takeaways from the webinar. Well, NMA empowers comparative effectiveness research. So, as we have seen, NMA is a powerful framework that lets us evaluate multiple treatments at once, even when direct comparisons are lacking. By integrating both head to head and indirect evidence, NMA creates a more complete picture of how different treatments perform. Next, essential tool for evidence based decision making. Today, HTA agencies, payers, and regulatory authorities rely heavily on NMA to inform clinical and reimbursement decisions, whether it's ranking interventions, supporting formulary listings, or supporting clinical guidelines. NMA is the standard. Flexible methodology supporting both frequentist and Bayesian frameworks. We explored how both frequentist and Bayesian approaches are used in practice, each offering unique benefits depending on the evidence structure and regulatory expectations. Next is advanced technique, enhance robustness and relevance. So Nathan highlighted methods like MAIC and MLNMR, which help bridge the gap between ideal clinical trial setting and real world populations, making evidence more applicable and defensible. And lastly, hands on implementation made accessible with R. So finally, Daniel demonstrated how R packages like NetMeta and multi-NMA can bring these concepts to life and how you allowing how you run the analysis, visualize networks, and produce reproducible treatment rankings. Thank you, and I will now pass it back to Sarah to facilitate any Q&A.
Thanks so much, Ankit. And we are going to jump right into the Q&A. We have lots of questions, which is wonderful. So if you guys are ready, I'm just going to push forward. Okay, so question for Ankit here. Is publication bias funnel plot a must in all NMA, and what is its relevance?
Okay, I can take that one. So, first of all, publication bias occurs when studies with statistically significant or positive results are more likely to be published than those with non significant or negative findings, right? So, this can distort the results of meta analysis because the studies with null or negative results might be missing, and so the overall effect could be overestimated. And so, funnel plot, so, in the absence of bias, the plot should uh, resemble a symmetrical inverted funnel. And asymmetry suggests possible publication bias, especially if small studies with negative or null results are missing, and only large positive result studies dominate. But is funnel plot a must? Well, not always, but funnel plots are useful and then they have some limitations. So, what they are good for? Well, meta-analysis with more than 10 studies, uh, that's good thing, and that's when the funnel plot helps, and then also detecting small study effects and asymmetry visually. And in terms of the limitations, the asymmetry doesn't always mean publication bias. It could also be due to poor heterogeneity or poor study quality, some random variation. But then, so there are some other methods to assess the publication bias, and one of them being Aggar's regression test. And so it's a formal statistical test for funnel plot asymmetry, and it's more objective than visual inspection actually. And then there is a Beck's test, and that's rank correlation method between effect estimates and variance. And then there are some other techniques like trim and fill method, P curve or U form analysis, and some others. Yeah, so there are some other methods out there. Yeah, and they have their own pluses and where ones to be used. So funnel plot, it's the good thing is that it's visual, useful when there are 10 studies, quick to check bias. Aggar's test is more statistical. It relies more on the formal test of asymmetry. Trim and fill is another statistical test, and it adjusts for the pool effect of bias, etc. I hope that answers your question.
Thank
Thank
you.
you.
Yeah, thank you for that. I know we are almost at time, everybody. So if you can stick around, we're going to just do at least one more question. But if you can't, that's okay. We'd love to get your feedback in the survey, but you can also come back and watch the recording later, and you'll get an email when that's ready. Okay, next question here. For Bayesian NMA, which one is a preferred package between GE, MTC, and multi-NMA, and why?
Okay, I think I can
Go
take
ahead.
it, right?
Yeah.
Okay. It massively depends on your familiarity with first R programming, and secondly, I would say with the complexity of Bayesian meta-analysis. For example, if you are conducting basic Bayesian network meta-analysis and you have a ton of data and you prefer a simple interface or are teaching NMA basics or something like that, and your computational resources are not so specialized or so sophisticated, I would opt to using GMTC package. And I'm personally more familiar with the multi-NMA package. But absolutely 100%, if you need advanced models like network meta-analysis with covariates or multivariate outcomes or network meta-regression or multi-level network meta-regression models, absolutely. And you care about faster convergence, precise posterior estimation, or even more robust diagnostics, I would say, I would opt to use the multi-NMA package. Both have useful vignettes, so you can access to them to learn how the package functions because you have the complete workflow. But in short, it depends. It depends on your familiarity with our programming and so on. Yeah.
Okay, thank you for that, Daniel. I did want to take this time to thank you all for your wonderful presentations, your insights in the Q&A. Thanks so much for being here, and yeah, great job. And in closing, we hope that you enjoyed this HealthEconomics.com webinar sponsored by ICON, and I hope that you enjoy the rest of your day. Thanks so much.